Hello and welcome to today's video lesson. It is on prokaryotes and eukaryotes. The learning goal for today is firstly to recognize the requirements of all cells for survival, and there's a few. We look at their energy sources, how they require matter, how they require simple nutrients, these important ones such as monosaccharides, disaccharides and polysaccharides, as well as various others such as amino acids and fatty acids, and how they remove wastes. We'll recognize that prokaryotic cells and eukaryotic cells have things in common and how that's said to be a feature of their evolutionary past. We will also just recall different structures and how those structures have different functions when looking at prokaryotes and eukaryotes, and how those various eukaryotic structures allow them to undergo more complex biochemical processes, such as how chloroplasts allow for photosynthesis, mitochondria allow for cellular respiration, and just various other organelles there. Again, how they remove wastes. We'll look at different features through an electron micrograph to be able to identify organelles. And lastly, compare the structure of prokaryotes and eukaryotes. So here, you can pause the video and just write really summarized versions of those learning goals before we jump into it. Cool, let's jump in. So an overview of where we're looking at right now. So every living thing we could think of as, well, it's either made up of cells or it's not made up of cells. If it's made up of cells, cells are the basis of every living thing. So we can think, well, there's two major types of cells. We have prokaryotic. They're relatively small compared to eukaryotic, and an example being bacterial cells. Eukaryotic cells make up every other living thing, so fungi to animal cells to plant cells. If we go the other way, we think about non-cellular organisms, then we have viruses, and it's this debate whether they're living or not. So this is a major division. Today we're going to zoom in on eukaryotes and prokaryotes, but focusing more on eukaryotic cells because they're a little bit more complex. Alright, so from 2.1 of the textbook, requirements for cell survival, the learning goal, as we already looked at, is to look at these various things. So what does recognize mean? It's quite a lower level cognitive verb, but it does require us being able to recall features. So let's get into it. The first one we see is energy. All cells require some form of energy. That is to grow, to reproduce, to move, and to carry out the maintenance and repairs needed for survival. Organisms can be classified on their ability to obtain energy. For example, autotrophs, as the word suggests, auto means they produce their own. It happens automatically within them. And an example being plants. Plants can obtain sunlight and convert that into chemical energy, as opposed to heterotrophs. And hetero means like of another. So heterotrophs obtain their energy from consuming other organisms or their dead remains. So after energy, we have matter. So every living cell is involved in synthesizing, that means building, larger molecules. These larger molecules are called macromolecules or biomolecules. And there are four types. We have carbohydrates, lipids, proteins, and nucleic acids. So autotrophs produce their own matter that they require, but heterotrophs, we get this from our diet. We also require gases such as oxygen. So we breathe in oxygen from the air, and that comes into our lungs. And so from our lungs, from the alveoli, these sacs at the end of our lungs, the oxygen diffuses out and into our blood, where it is then transported all around the body where the oxygen then moves from the blood into the cell from an area of high concentration to an area of low concentration. There is low oxygen in the cells because that's what our cells use. The same but opposite process happens with carbon dioxide. Our cells produce CO2 that goes from our cells into the blood and from our blood into our alveoli, and then we exhale that. This occurs in the gills of fish, the lungs of mammals, birds, and reptiles. So nutrients, cells use carbohydrates to obtain energy. So these are used to build up larger macromolecules. Simple carbohydrates are glucose molecules, which we obtain from our diet, and those simple glucose molecules, like the simplest being monosaccharides, can be built up into larger disaccharides. You'll see in the textbook when two monosaccharides come together, but when two or more disaccharides come together, they produce more complex forms of carbohydrates, more complex forms of sugars, and that is polysaccharides. As well as this, nutrients that we require are glucose, starch, and cellulose. So glucose, when we obtain it from our diet, if it's not used immediately, it's stored in the body of mammals, of animals, of us as glycogen. So we require glucose, that's converted to glycogen. But plants, they produce cellulose, and that is important because it becomes a component of plant cell walls. It's a lot harder, it's a very complex sugar compared to other more simple ones. And as I said before, starch, it's an important energy storing polysaccharide in plants. So like humans convert glucose into glycogen, plants convert their sugars into starch to be stored. A variety of other substances required for proper functioning of cells include the following terms from your textbook. And so there are fatty acids. They're a type of organic acid that combines with glycerol to form a fat molecule. And glycerol is a molecule that combines with fatty acids to form a fat molecule. We also require amino acids. They are a nitrogen-containing compound that is the building block of proteins. Year 12s, you might think back to last year. So proteins are so important because they make us up. So we require amino acids, as well as triglycerides. They're a simple lipid formed by linking glycerol with 3 fatty acids, so when they come together. As well as this, we also require ions, and that is IONS, and they're charged molecules, and they include potassium, magnesium, sodium, chloride, phosphate, iron, and of course water is essential because water is vital in our body as it allows for chemical reactions to take place. And chemical reactions is how things get done, is how we produce energy, it's how we do everything that we need to do, basically. So nutrients is very important. And we've spoken about these ones. We've just spoken briefly about these ones. Remember, the cognitive verb is to recognize. So we don't have to explain as such. And lastly, to survive, cells need to remove wastes. And so various wastes include carbon dioxide. It's a byproduct of cellular respiration. I'll speak about that a little bit more later. Even oxygen and water are byproducts. Metabolic heat, so as we're breaking down sugars into energy, heat is released, so our body needs to be able to let off heat. But the one I want to speak about a little bit more is this. Ammonia is a product, it's a byproduct of metabolizing sugars into energy, but it's actually extremely toxic in humans. So we need to do something about that. And what we do is we convert it into a less toxic compound, and that is urea. Urea is then diluted and excreted in urine in mammals. But in other organisms, it's converted into what is called uric acid or uric acid crystals. This is how they excrete their ammonium waste. And it's this waste product produced by desert animals such as birds, reptiles, and insects. Basically, when they don't have much water, they need to excrete it in another less toxic form. And that is uric acid. So this is an overview of 2.1 in your textbook, talking about the requirements for cell survival. So we can recognize how these various things are all needed, then we're doing a good job. Okay, moving on. So prokaryotic and eukaryotic cells. So we have these two major types of cells, as I have said before. Prokaryotic cells, the most general difference between them is that they lack a nucleus, and that's what even the names of these cells suggest. Whereas eukaryotic cells, they do have a nucleus, and this is where the genetic information is contained. So two types of things make up prokaryotes. They are bacteria and archaea. Eukaryotic cells make up pretty much every living thing that you can think of: plants, animals, humans, fungus. Bacteria are much smaller, prokaryotes are much smaller, and these images are not to scale. Say, if this were a prokaryote, it would probably be about the size of a mitochondrion in retrospect to a eukaryotic cell. Although prokaryotes are much smaller and simpler, they do still have similarities with eukaryotes, and one of them is they both contain DNA. In prokaryotes, it's said to be circular DNA, whereas in eukaryotes, that DNA is contained within the nucleus instead of just floating around freely in the cytoplasm. They also have cytoplasm or cytosol. It's just this solution that everything else is kind of suspended in. Even though it's not free floating, there is actually skeletons within these organelles within a eukaryote. Anyway. Then there's also ribosomes. Both prokaryotes and eukaryotes contain ribosomes, and they're really important for making things, making proteins. So when we look at eukaryotic cells, we have all these organelles, and that word is Latin, and it just means little organs. The reason why eukaryotes are more complex and can do more things and make more complex organisms when they even come together is because of these membrane-bound organelles. It's like little environments within each one of these that allow them to specialize and perform a particular function. If we were to look at the history of prokaryotes and eukaryotes, they suggest in endosymbiotic theory, which you'll see in your textbook, to propose how eukaryotics came to be. They believe that one prokaryote maybe once engulfed another, and so that's how we have like this little kind of prokaryotic feature within what may have been once a larger prokaryotic feature. And then over millions of years, these features just evolved into having their own function and now performing altogether. That is the endosymbiotic theory. However, it is also completely unprovable in the way we cannot test it, we cannot repeat it, we cannot observe it, but it is widely accepted in textbooks and universities, and so thus taught. Prokaryotic cells are simpler and smaller, and they make up, as I said, bacteria and archaea, but eukaryotes are much larger and more complex, and so we're going to focus on them a fair bit more in this video lesson. So, just to recap what we spoke about, the differences and similarities. So prokaryotic cells do not have a nucleus. They lack some organelles, they're not found in humans, and they're always unicellular, like organisms that are just made up of one cell, such as a bacterium. They reproduce by binary fission, so that just means like by replicating and dividing. And they contain circular DNA, as opposed to eukaryotic cells having a nucleus, having organelles found in humans, often come together to make multicellular complex organisms such as you and I, and they reproduce by this process called mitosis and meiosis, and we learned about that in Unit 3 and 4. And DNA, which is passed on, it's ancestral. So if we focus on eukaryotic cells, here is a drawing of a eukaryotic cell, and it is an animal cell as opposed to a plant because it does not have a cell wall, and it does not have what I can see on here as chloroplast, or no, a large vacuole. It's quite small. So it's an animal cell, and there are quite a number of organelles that make up an animal cell, and it looks pretty boring when you just look at it, but these cells are incredibly complex. And if you check out some videos online of animations, there's things that are all performing their own functions, and there's like things that are literally walking inside the cell. They're constantly moving and alive, and so incredibly complex. It's crazy. But for the sake of getting an introductory understanding of eukaryotic cells, we just look at these simple diagrams to get our heads around the main features and what they do. So I'm going to talk about each organelle and what they do. And we can start over here with the nucleolus, and it's this feature within the nucleus, and it's where ribosomes are assembled. The nucleus, though, was one of the first organelles ever discovered, and it contains the genetic material of the cell, which is DNA. That DNA determines what kind of cell it's going to become, a muscle cell or a neuron, and it also controls the cell by regulating just the chemical reactions and the functions, everything that's going on inside of it. So we could call it like the control center of the cell, or the brain. Year 12s, you would remember that the nucleus is this like spherical feature with all these little holes in it, pores, and that's important in transcription and translation. But don't worry about that, year 11s. We'll cover that next year. We also have ribosomes, and ribosomes are kind of represented often as these little dots, but they're a lot more complex than that as well. If we zoomed in, they've actually got like a few compartments, and they play a crucial role in survival of anything. They're essential for creating proteins. Proteins make us up. Then we have the vacuole, and the vacuole is this storage compartment. In plants, it is really large as it stores water and sap. We can look at the endoplasmic reticulum and see that there's a rough endoplasmic reticulum and a smooth. What makes it rough? Well, it's the ribosomes that attach to it and give it this rough look. And ribosomes, we said, are essential for making proteins. So the endoplasmic reticulum we can think of as this big kind of factory, this folded membrane full of ribosomes on the outside, which is then just producing proteins. So that's like a factory. And if we think, well, what happens to all those things that are made? Where do they go now? They go to the Golgi apparatus, and the Golgi apparatus kind of snazzes them up a bit, adds a few things, puts some carbohydrates on, and then it packages it and sends it to where it needs to go, either in the cell or outside of the cell. It's kind of like the shipping or the postal service of the cell. We also have microtubules and microfilaments. So this is like the skeleton of a cell, and it just provides a bit more structure. Microtubules are these larger kind of features. So if you think about like a bridge, it has those big poles, but then microfilaments are like the wires coming off that central pole, and they just provide scaffolding and structure for the cell. As I said before, there's also a smooth ER, endoplasmic reticulum, because it does not have ribosomes attached, but it's still important in manufacturing things. Not proteins, but lipids. And we know lipids are really important for our cell membrane. It also produces carbohydrates and steroids, these hormones. And the smooth endoplasmic reticulum is essential in breaking down toxins in our body. So alcoholics would have a lot larger surface area, more volume of smooth endoplasmic reticulum to break down that constant supply of alcohol. We also have mitochondria, and you might know mitochondrion as being the powerhouse of the cell, and basically this is where we generate energy. Whenever we talk about energy in a cell, that's in the form of ATP. It is this folded membrane. It's got all these folds inside because it allows for more surface area, and more surface area just means more area to get things done, for reactions to occur. And it's really important in the process of cellular respiration. And that's pretty much when we have sugar from our diet, oxygen from the air, and we take all of that, and in this magical process we create energy for our cells and for our body. Then we have the cytosol, or the cytoplasm. It's this solution that kind of fills the space. It is more complex than we first thought, but we still don't know exactly what it does. We also have centrioles, and they're important in just creating positioning within the cell. They kind of structure where the nucleus will be, where other things will be, and they're also really important in cellular division. They pull different parts of the cell to each polar opposite end, and eventually that cell will then split. Well, not on here, but when talking about eukaryotic cells, we can talk animal cells, but we can also talk plant cells. Plant cells are similar, but they also have a few more extra features that provide, number one, a cell wall gives the plant a more rigid structure because plants don't have a skeleton, so they require a cell wall for that. And also, as you will read about in the textbook, we also have what are called plastids. And plastids are these double membrane features that can actually interchange between different types of plastids depending on what the cell needs. So an example of a plastid is a chloroplast, and a chloroplast is important for cellular respiration. So, photosynthesis. We also have, as you will read in the textbook, tenosomes, and they produce tannins. It's this compound produced by plants, and the most abundant common form of tannin are polyphenols. And polyphenols are compounds that protect plants from attack. It's like this chemical barrier. Attack from herbivores or pathogens. So there are so many things we just spoke about, and I think one of the best ways to learn this is just creating flashcards. So write on one side ribosomes, flip it, and then either have a drawing, a definition, what they do. Just simple keywords. Keep it simple. So that is that. And if I move on, one of the learning goals was to be able to identify features through looking at a microscope. And these are taken using not the ocular light microscopes that we use in the classroom. They wouldn't provide such definition, but electron microscopes where they fire electrons at images, and the way they bounce off, and that creates this detailed image. But I'm going to exit the slideshow now to show you guys this website, and it's called BioNinja. It's a really good source of information. I will include the link to this website in the description of this video so you can check it out yourself. But it's just this electron microscope, images of it, and it shows us like prokaryotic cells. It's a bit interactive too, how you can select like the flagella, and the flagella are these little tails that come off prokaryotic cells. But I want to focus on eukaryotic cell features, and here you can see that eukaryotic cells, they have a nucleus, and here in this animal cell, it's massive. It's also over here in this plant cell, and there's the nucleolus. We know eukaryotic cells have endoplasmic reticulums. It's this big factory, isn't it? These folds that create, through ribosomes, proteins. And we also know that we have mitochondria in eukaryotic cells. We also need them in plant cells. Like chloroplasts create the energy, but we need mitochondria to still like break that down to use it. We also have Golgi apparatus. It's this, again, folded membrane, the post office of the cell. Chloroplast, it's not going to be in an animal cell. And here we can see that they're quite large. They take up a lot of space. Cell wall, again, animal cells don't have a cell wall, but we can see plant cells do. Check out the size of this vacuole. So it's so small. And yeah, there it is. So organelles, we have a Golgi apparatus. So we're zoomed in quite a fair bit here because we saw that it's not massive. Chloroplasts are quite big. Mitochondria, we can see that from all these folds within it, creating surface area. And endoplasmic reticulum with all these ribosomes on them. So mitochondria cells, if you would find them in neurons and muscle cells where like they require a lot of energy. Endoplasmic reticulums are these vast networks. They're often used in secreting activities. So you'll find them in like exocrine glands, glands that produce and release hormones. Lysosomes, they are the kind of the organelle that breaks things down. They contain digestive enzymes, and so phagocytes are white blood cells, and white blood cells are the important part of our immune system that engulf and digest and break down foreign or harmful materials such as bacteria. So lysosomes would be rich in phagocytes. In chloroplasts, we know that would be in plant cells. So that again is BioNinja, if you want to check that out yourself. So if we get back into this though, and we move on, just to look back at all the learning goals we just covered, we can recognize, recall, understand, and compare. So there was a lot of information in this, I know. But if you look at the cognitive verbs, they're not massive. We don't have to necessarily write essays on each one of those things. So, moving on. Homework. Chapter 2.1 to 2.6. That's what I've taken and condensed into this tiny 30-minute video. I've said read it. I know that you could spend a lot of time doing so. So give it a go. Even if you need to skim or just pull out the key terms or look and make sense of the images. There's a cool little section in there where it does focus on images and identifying them under a microscope. Give it a go. Create flashcards. So, alright, the create for this week is to either create flashcards of these organelle terms, all those lysosomes, mitochondria, nucleus, nucleolus, endoplasmic Golgi apparatus. Create something to help you learn that. Or a descriptive model of a eukaryotic cell. That can be physical, it can be virtual, it can be edible. Create something this week. And that will be due the last lesson of week 4. So support, check out the Amoeba Sisters video. It takes you on a tour of the cell. Uses great visuals, simple language, and the link will be in the description of this video. Extension, you can find in your textbook just these cool kind of questions that it poses towards you. And it says the development of the cell theory. I'll let you read that yourself if you want to check it out. So you can pause the video here, write what you need to do down. And just to recap now, everything that we've spoken about, prokaryotes means like before they were complex, before there was a nucleus, whereas eukaryotes means like true, they do have a nucleus. And eukaryotes are fungi, animals, plants, protists, but prokaryotes are just archaea and bacteria. Again, another quick comparison. They both have a cell membrane, both have cytoplasm, DNA, ribosomes, but when it comes to organelles, they're a lot more complex in eukaryotic cells. So a few of those organelles we covered: the cytoplasm, it's the jelly-like substance; the Golgi apparatus, it's the post office of the cell. We have lysosomes, which come and engulf and digest in the process of phagocytosis. Phagocytes are an example of this, that's white blood cells. Oh, by the way, interesting fact, they're also called like the suicide organelle, because when a cell has come to its end, its life, it's done its purpose, it is actually the lysosome that bursts and releases all its digestive enzymes, and that just like kills the cell. The mitochondrion, it is the mighty ATP producers of the cell. Then we have the nucleus, which contains all the genetic material. We have ribosomes, which produce proteins. We have the rough endoplasmic reticulum, because it contains ribosomes, rough with ribosomes, and is the protein assembly line of the cell. The vacuole, which is the storage compartment, it's large in plants for sap and water. We do have vacuoles in humans as well, just smaller. And then lastly, we have the chloroplasts, which are of course only in plants. They produce glucose, sunlight into energy. Pretty cool. But you have done well. Well done. One more thing. Cellular respiration, that was just the process of taking sugar, glucose, oxygen, and producing energy. Byproduct is carbon dioxide and also water. It's actually the exact opposite, back to front process of photosynthesis. But anyways, here we are. That's all, folks. Congratulations on making it to the end. I hope that this video has been informative and helpful, and along with the discussions and activities in class and your readings, that you can get a firm understanding on everything prokaryotes and eukaryotes. God bless you guys, and I will see you in class.